Hello, everyone, and welcome. I'm glad you're here today. We're about to explore an exciting topic in biology. The slide shows the title, The Cell, the Unit of Life. Today, we'll look at what cells are, why they are so important, and how they form the building blocks of all living things. So, let's get started and discover the fascinating world of cells together. Let's take a closer look at the structure of an animal cell. Understanding this is key to grasping how cells function and interact within living organisms. We'll explore various components, starting with the centrosome and centrioles, which play a vital role in cell division. Moving on, chromosomes are essential for genetic information storage and transfer. The mesosomes, although less prominent in animal cells, help us compare different cell types. The overall cell, of course, is the fundamental unit of life, and by studying its parts, we better understand biological processes. As we go along, we'll also look at the cytoskeleton, which provides shape and support, and the various organelles like the nucleus, endoplasmic reticulum, and mitochondria that carry out specific functions. This slide sets the foundation for understanding how complex yet organized animal cells are, tying into our larger theme of cellular structure and function. In 1965, Robert Hooke made a fascinating observation. He was examining slices of cork taken from the bark of a tree using a simple magnifying glass. What he saw were tiny, box-like structures that looked like compartments. These small boxes or chambers were separated from each other by thin walls or partitions. Hooke decided to call each of these boxes a cell. It's interesting to note that the cells he observed were actually dead plant cells. This discovery was really important because it marked the first time anyone had identified these basic units of life, which would later become fundamental to our understanding of biology. So, in essence, Hooke's work laid the groundwork for the study of cellular structure and helped us realize that all living organisms are made up of cells. All organisms, whether they are plants, animals, or microbes, are made up of one or more cells. The cell is really the fundamental building block of all living things. It's where the essential activities of life happen, and it provides structure and function to the entire organism. An important concept to remember is that new cells come from existing cells. This idea is summed up in the phrase omnis cellula cellula, which means all cells come from cells. So, cells don't just appear out of nowhere, they multiply from pre-existing cells. The cell is actually the smallest unit of life that can carry out all necessary functions. It's like the minimal working unit that keeps an organism alive and functioning. The development of cell theory was a major milestone in biology. It was first formulated by Matthias Schleiden and Theodor Schwann, who recognized that all living things are made of cells. Later, Rudolf Virchow added an important piece to the puzzle: the idea that all cells originate from pre-existing cells, which really cemented the theory. So, in summary, cell theory tells us that all organisms are made of cells, that the cell is the basic unit of life, and that new cells come from existing ones. These principles help us understand the structure and function of all living things, tying into the bigger picture of biology's fundamental concepts. Let's talk about prokaryotic cells. These are a fundamental type of cell on Earth, and they are quite simple compared to eukaryotic cells. One of the key features is that their DNA is not covered by histones, so it's not enclosed in a nucleus. Instead, the DNA is free-floating within the cell. Examples of prokaryotic cells include bacteria and blue-green algae, which are some of the earliest forms of life. In fact, they are considered the first type of cell on Earth. These cells are very small, typically ranging from 1 to 10 micrometers, which helps them survive in a variety of environments. Another important point is that prokaryotic cells lack membrane-bound organelles like mitochondria or chloroplasts, which are found in eukaryotic cells. This simplicity is part of what makes them so efficient and adaptable. Some common examples are bacteria, mycoplasma, and blue-green algae. They also reproduce asexually, primarily through processes like budding and fission, allowing quick population growth. Lastly, as the name suggests, these cells have no nucleus. The term prokaryotic comes from Greek, where pro means primitive and karyon means nucleus, highlighting their early evolutionary status and simple structure. Let's take a closer look at eukaryotic cells. Generally, these cells range in size from about 10 to 100 micrometers, which is a bit larger than prokaryotic cells. You'll find that they include fungi, protists, plant cells, and animal cells. One of the key features that sets eukaryotic cells apart is the presence of membrane-bound organelles. These specialized structures inside the cell perform specific functions, making the cell more organized and efficient. It's also important to note that all these organisms, except bacteria and blue-green algae, are eukaryotic. They all have a true nucleus, which is a defining characteristic. The word eukaryotic actually comes from Greek, where EU means true and karyon means nucleus. So essentially, we're talking about cells with a true, well-defined nucleus. Understanding these features helps us see how complex and organized eukaryotic cells are, especially compared to simpler cells like bacteria. They are a fundamental part of the diversity of life we're exploring in this presentation. Organisms that are made up of many cells are called multicellular organisms. For example, a tree or an elephant. These organisms have complex structures because their bodies are composed of numerous specialized cells working together. On the other hand, organisms made up of only one cell are called unicellular organisms. Think of amoebas or paramecia. They are single-celled but still alive and functioning just fine. Cells are really the basic building blocks of all living things. No matter how big or small an organism is, it all starts at the cellular level. Some organisms, like bacteria or certain protists, are made up of just a single cell, while others, like trees, elephants, or humans, are made up of billions or even trillions of cells. So, understanding cells helps us grasp how life is organized, from tiny single-celled creatures to incredibly complex multicellular beings. This foundation is key to understanding the diversity and structure of living organisms overall. Now, let's take a closer look at the different shapes that cells can have. Cells can be branched, which means they have a kind of tree-like structure with multiple extensions. Some cells are spindle-shaped, kind of like an elongated oval, which allows them to move easily through narrow spaces. Other cells are elongated themselves, stretching out in a particular direction, which can be important for their function. Then, we have round and spherical cells, which are more compact and often serve different roles in the body. It's also interesting to note that some cells are constantly changing their shape, like amoebas or certain white blood cells in humans. This shape-shifting ability helps them move and adapt as needed. Overall, the shape of a cell is closely linked to its function and environment. Different shapes enable cells to perform specific tasks efficiently, which ties into the larger theme of how form follows function in biology. Let's start by looking at the range of cell sizes in living organisms. The largest cell is actually the egg of an ostrich, which measures about 17 centimeters by 13 centimeters. That's quite big compared to most cells we're used to thinking about. On the other end of the spectrum, the smallest cell is from a type of bacteria called PPLO, which is only about 0.1 to 0.5 micrometers in size. This huge difference in size, from a fraction of a micrometer to over 17 centimeters, shows just how diverse cells can be. Cells in living organisms can be as tiny as a millionth of a meter, which is called a micrometer or micron, or as large as a few centimeters. This variation is really important because it reflects the different functions and structures of these cells. Understanding these size differences helps us appreciate the incredible diversity of life at the cellular level and how cells are adapted to their specific roles. Let's take a closer look at the cell membrane, or plasma membrane. It contains two main types of proteins: peripheral proteins, which are attached to the membrane surface, and integral proteins that span across the membrane. Along with these proteins, you'll find cholesterol, glycolipids, and glycoproteins embedded within or associated with the membrane. The primary function of the membrane is to act as a semi-permeable barrier. This means it allows certain molecules to pass through while blocking others, which is crucial for maintaining the cell's internal environment. According to the fluid mosaic model, the membrane's lipids are not rigid. They are semi-fluid, which enables lateral movement of the proteins within the bilayer. This fluidity is essential because it allows proteins to move and function efficiently, adapting to the cell's needs. In erythrocytes, or red blood cells, the membrane composition is roughly 52% protein and 40% lipids. This specific balance plays a vital role in the cell's flexibility and functionality. This model was proposed by Sanger and Nicholson in 1972 and has since been fundamental in understanding cell structure. Finally, the outer membrane of the cell controls what enters and exits the cell, ensuring substances move in accordance with the cell's requirements. It's this selective permeability that keeps the cell functioning optimally. Now, let's look at some examples of transport mechanisms. First, we have the Na plus K plus at pace pump. This is a classic example of active transport. It moves molecules from a lower to a higher concentration, and it requires energy in the form of ATP to do so. This process is crucial for maintaining cell function and homeostasis. On the other hand, we have passive transport, exemplified by diffusion. Diffusion involves molecules moving from an area of higher concentration to an area of lower concentration, and importantly, it does not require energy. This process is driven purely by the concentration gradient. Understanding these mechanisms helps us see how cells control the movement of substances across their membranes, which is fundamental to their survival and function. Now, let's look at mesosomes and their role in the cell. They assist in respiration and secretion processes, which are vital for the cell's energy production and waste removal. Additionally, mesosomes increase the surface area of the plasma membrane, providing more space for enzymes and other proteins necessary for metabolic activities. This expansion enhances the efficiency of these processes. Furthermore, mesosomes are involved in cell wall formation, helping to synthesize and organize the cell wall components. They also play a crucial role during DNA replication, ensuring that genetic material is accurately copied and evenly distributed to the daughter cells during cell division. You can see that mesosomes often appear in the form of vesicles, tubules, and lamellae. These are essentially extensions or invaginations of the plasma membrane into the cell. They are formed by the plasma membrane extending inward, creating structures that facilitate various cellular functions. In essence, mesosomes are important membranous structures that support several key activities within prokaryotic cells, linking membrane dynamics to critical processes like reproduction and metabolism. Let's talk about the cell wall, starting with algae. The cell wall in algae is composed of a mix of different substances, including cellulose, galactans, mannans, and minerals like calcium carbonate. These components give the cell wall its strength and structure. Now, when we look at plant cell walls, they are primarily made up of cellulose, but also include hemicellulose, pectins, and proteins. These materials work together to provide the plant cell with rigidity and support. The key point here is that the cell wall acts as a protective barrier. It helps prevent undesirable macromolecules from entering the cell, which is crucial for maintaining cellular integrity. It's important to remember that the cell wall is a non-living material, yet it plays a vital role in plants, fungi, and bacteria by surrounding the plasma membrane. This layer not only offers protection from mechanical damage and infection, but also helps maintain the shape and support of the cell. So, in summary, the cell wall is essential for providing structure, support, and defense, making it a fundamental part of the overall health and function of these organisms. Now, let's take a closer look at some of the modifications of the cell envelope that are crucial for its functions. First, we have mesosomes, which are essentially extensions of the plasma membrane. They can take various forms like vesicles, tubules, and lamellae, and are thought to play roles in processes like cell division and respiration, although their exact functions are still a topic of discussion. Next, there's the capsule, which is a thick, tough sheath made of glycocalyx. This structure helps protect the cell from environmental threats and can also be involved in adhesion to surfaces, which is important for pathogenicity in some bacteria. On the other hand, we have the slime layer, which is also glycocalyx, but in a looser, more diffuse form. It acts as a protective barrier and aids in the movement of bacteria by reducing friction. Moving on to the layers of the cell wall, we see the secondary wall, which is the innermost layer. It becomes lignified in sclerenchyma and vessel elements, providing additional strength and rigidity. In the endodermis, it's suberized, helping in selective permeability. The primary wall, present in young cells, is capable of growth and is mainly composed of cellulose and hemicelluloses. This flexibility allows the cell to expand during growth. Finally, the lamella is the outermost layer, made mostly of calcium pectate. Its main role is to hold neighboring cells together, ensuring structural integrity within tissues. All these components work together to give the cell its protective, structural, and functional characteristics, which are essential for the organism's overall health and development. Now, let's talk about the cytoplasm. The cytoplasm is the portion of the protoplasm that lies inside the cell membrane, but outside the nuclear membrane. Think of it as the jelly-like substance that fills the inside of the cell, providing a sort of cushion and medium for all the cell's activities. It's important to note that this gel-like fluid contains many vital components, including hereditary material, which is essential for the cell's functions and inheritance. Essentially, the cytoplasm is like the bustling interior of the cell, supporting and suspending the organelles and enabling the cell to operate smoothly. Understanding the cytoplasm helps us appreciate how cells maintain their structure and carry out their life processes efficiently. Let's take a closer look at the nucleus and its components. Chromatin, which is found inside the nucleus, contains DNA along with some basic proteins called histones, as well as non-histone proteins and RNA. Each chromosome has a primary constriction known as the centromere, and on either side of the centromere, you'll find structures called kinetochores, which are crucial during cell division. Speaking of which, during cell division, this complex chromatin network condenses into the visible chromosomes we often think about. Now, the nucleus isn't just a simple container. Nuclear pores are essential because they provide passages for the movement of RNA and protein molecules between the nucleus and the cytoplasm. Inside the nucleus, you find the nucleoplasm, which contains the nucleolus and chromatin. The nucleolus is particularly important because it's the site where active ribosomal RNA synthesis occurs. Usually, a cell has only one nucleus, but interestingly, some mature cells lack a nucleus altogether, such as erythrocytes in many mammals and sieve tube cells in vascular plants. The nuclear envelope surrounds the nucleus and consists of two parallel membranes separated by a space called the perinuclear space. The outer membrane is often continuous with the endoplasmic reticulum and has ribosomes attached to it, which plays a role in protein synthesis. The material inside the nucleus that stains with basic dyes was named chromatin by Fleming, highlighting its importance in genetic material. During the interphase of the cell cycle, the nucleus contains nucleoprotein fibers called chromatin, along with a nuclear matrix, and one or more spherical structures known as nucleoli. Think of them as the cell's ribosome factories. This overall structure and organization of the nucleus was first described by Robert Brown, which really helped us understand how genetic information is stored and managed within the cell. So, in summary, the nucleus is a highly organized, dynamic organelle that plays a central role in genetic regulation and cell function. All right, so let's talk about chromosomes, which are really fascinating structures. They are responsible for the inheritance of characters, or genes, from one generation to the next. In other words, they carry the genetic information that determines traits. Chromosomes are made up of DNA, which is the molecule that holds all this genetic information. You'll find chromosomes inside the nucleus of a cell, which acts as the control center of the cell. Understanding chromosomes helps us see how traits are passed down and how genetic information is organized and maintained through generations. This is a key piece of the bigger picture of genetics and inheritance. Let's take a closer look at the mitochondria, which are often called the powerhouses of the cell. These structures contain a single circular DNA molecule, a few RNA molecules, and ribosomes, specifically 70S ribosomes, which are essential for protein synthesis. Mitochondria multiply through a process called binary fission, similar to how bacteria reproduce, ensuring they can meet the energy demands of the cell. They play a crucial role in controlling the levels of water and other materials inside the cell, helping maintain a stable environment. Additionally, mitochondria are responsible for recycling and breaking down proteins, fats, and carbohydrates, which is vital for cell health. Most importantly, they carry out cellular respiration, the process that produces energy in the form of ATP, making them the true energy producers of the cell. Structurally, mitochondria are rod-shaped or spherical, with a double membrane that encases their inner workings. This specialized structure supports their role in energy production and metabolic processes. So, in essence, mitochondria are integral to both the energy supply and overall functioning of the cell, linking closely to the theme of cellular efficiency and specialization. Now, let's take a closer look at plastids. These are large organelles that are characteristic of plant cells. There are three main types of plastids, each with a specific function and pigment. First, we have chloroplasts, which are perhaps the most well-known. They contain chlorophyll, the pigment responsible for the green color of plants. Chloroplasts are crucial because they are the sites of photosynthesis, where plants convert sunlight into energy. They also contain DNA and 70S ribosomes, which are essential for their function. Next, we have chromoplasts. These plastids contain carotenoids, which give color to various parts of the plant. Think of yellow, red, and orange hues seen in fruits and flowers. These colors are important for attracting pollinators and seed dispersers, playing a key role in plant reproduction. Finally, leucoplasts are involved in storing products like starch, oils, and fats. They are non-pigmented and serve as storage sites, providing energy reserves for the plant. Understanding these plastids helps us see how plants are adapted to perform photosynthesis, attract pollinators, and store energy efficiently, all vital to their survival and our ecosystem. Let's take a closer look at the endoplasmic reticulum, or ER. There are two types, the smooth ER and the rough ER. The smooth ER, as the name suggests, lacks ribosomes on its surface, and its main role is in lipid synthesis, so it helps produce fats and other lipids essential for the cell. On the other hand, the rough ER has ribosomes attached to its outer surface, and these ribosomes are key players in protein synthesis and secretion. The rough ER also acts as a sort of cellular highway, providing a network of membranes that gives the cell its structural framework and helps move materials around efficiently. Both types of ER are crucial for maintaining the cell's overall function and health, working together to produce and transport vital molecules. This interconnected system really highlights how dynamic and organized the cell is, aligning with our broader theme of cellular complexity and efficiency. Ribosomes are fascinating structures because they are essentially the sites where proteins are made. They are present in the cytoplasm, floating freely or attached to the endoplasmic reticulum, and they play a crucial role in translating genetic information into functional proteins. When we look at prokaryotic ribosomes, they are classified as 70S, which means they are made up of two subunits, a 50S large subunit and a 30S smaller subunit. This combination is a key feature that distinguishes prokaryotic cells from eukaryotic ones. Eukaryotic ribosomes, on the other hand, are slightly larger, classified as 80S, consisting of a 60S large subunit and a 40S small subunit. The differences in ribosome size and structure are important because they relate to how these cells produce proteins and how antibiotics can target bacterial ribosomes specifically. Importantly, ribosomes are composed of RNA and proteins, but they do not have a membrane around them. This makes them unique compared to other organelles, emphasizing their role as molecular machines rather than membrane-bound structures. So, in summary, ribosomes are essential for protein synthesis, present in all cells, and their structural differences reflect the diversity between prokaryotic and eukaryotic organisms. Now, let's take a closer look at the Golgi complex. Its main function is to perform the packaging of materials that the cell needs to deliver. These materials can be targeted either to intracellular destinations within the cell or secreted outside of it. Essentially, the Golgi acts as the cell's shipping department, ensuring everything gets to where it needs to go. Structurally, the Golgi consists of a series of cisternae, which are stacked parallel to each other. Think of them as a stack of flattened sacs, all arranged neatly in a row. These sacs are sac-like structures, and they are stacked one above the other, creating a layered, organized system. One of the key roles of the Golgi is to store materials produced by the cell. That's why it's often called the storehouse of the cell. It keeps these materials safe and organized until they are ready to be transported to their final destinations. Overall, the Golgi complex is crucial for processing, packaging, and distributing the proteins and lipids that the cell needs to function properly. It's a central hub in the cellular logistics system, maintaining order and efficiency in material trafficking within the cell. Now, let's talk about lysosomes. Their main function is intracellular digestion, which means they break down waste materials and cellular debris inside the cell. These structures are membrane-bound vesicles that are formed through a process called packaging in the Golgi apparatus. Essentially, the Golgi sorts and pinches off these vesicles to create lysosomes. Inside, they contain hydrolytic enzymes. These are special enzymes that break down complex molecules. What's particularly interesting is that these enzymes are active in an acidic pH, which helps optimize their function. Because of their destructive potential, lysosomes are sometimes called the suicidal bag, a vivid way to remember that they can digest cellular components if not properly controlled. So, in the context of our overall theme of cellular maintenance and function, lysosomes are vital for keeping the cell clean and healthy by disposing of old or damaged parts. They play a key role in cellular homeostasis by recycling materials and ensuring the cell functions smoothly. Let's take a closer look at vacuoles, which are membrane-bound spaces within the cytoplasm, known as tonoplasts. These structures are essentially storage sacs that hold water, cell sap, excretory products, and other materials that the cell doesn't need right now. It's interesting to note that in plant cells, vacuoles are usually quite large, often taking up a significant portion of the cell's interior. This size helps the plant maintain turgor pressure, which is crucial for maintaining the plant's structure. Another key point is that vacuoles play an active role in transporting ions and other materials into themselves, even against concentration gradients. This means they help regulate the cell's internal environment by moving substances in and out efficiently. This transport process is vital for overall cell health and function, especially in plants where vacuoles contribute to processes like nutrient storage and waste disposal. So, in the context of cell structure and function, vacuoles are more than just storage bins. They are dynamic, essential components that support the cell's stability and homeostasis. Now, let's look at slide 25, which focuses on cilia and flagella. These structures are fascinating because they are covered with plasma membrane, just like the rest of the cell. This means they are not exposed directly to the cell's interior, maintaining a protective barrier. When comparing them, flagella are generally longer than cilia, which is an important distinction. Cilia are smaller and work much like tiny oars, helping the cell move or assisting in moving fluids around the cell. This movement is crucial for many physiological processes. Overall, both cilia and flagella are hair-like outgrowths of the cell membrane, and they play key roles in locomotion and cell movement. Understanding their structure and function helps us appreciate how cells interact with their environment and perform essential tasks. Now, let's look at the centrosome and centrioles. The centrosome is a crucial organelle in cell division because it produces the spindle apparatus, which is essential for pulling chromosomes apart accurately. Inside the centrosome, you'll find two cylindrical structures called centrioles. These centrioles are surrounded by an amorphous, gel-like material known as paracentriolar material. The centrioles also play a key role as they form the basal body of cilia, or flagella, which are structures involved in cell movement and sensory functions. So, in essence, the centrosome acts as the organizing center for microtubules during cell division, while the centrioles have additional roles in forming the structures that help cells move or sense their environment. Understanding these components helps us see how cells coordinate division and movement, which ties into the larger theme of cellular organization and function. Let's take a closer look at how chromosomes are characterized by the position of their centromere. First, some chromosomes have a terminal centromere, which means the centromere is located at the very end of the chromosome. These are called telocentric chromosomes. Sometimes, you'll notice that certain chromosomes have non-staining secondary structures at a consistent location, giving the appearance of small satellite fragments. These are known as satellites. Moving on, we have chromosomes where the centromere is closer to one end, but not at the very end. These are called submetacentric chromosomes. The centromere here is situated near the end, but not quite at the tip. Then, there are acrocentric chromosomes, where the centromere is positioned much nearer to one end, creating a distinct long arm and a very short arm. Finally, the most central position is seen in metacentric chromosomes, where the centromere is roughly in the middle, resulting in two arms of approximately equal length. Understanding these variations is crucial because the position of the centromere influences chromosome behavior during cell division and helps us classify different chromosome types across species. Let's take a closer look at the cytoskeleton and its key components. First, we have the forms that scaffold the chromatin, acting like a basket around the nucleus, providing structural support. Next, there's the intermediate filaments, which are non-contractile hollow filaments made up of acidic proteins. These filaments help maintain cell integrity and resist mechanical stress. Moving on to microfilaments, these are composed of actin and myosin, which are contractile proteins. They form solid, unbranched rod-like fibrils that are essential for cell movement and shape changes. Lastly, we have microtubules, made of alpha and beta tubulin subunits. These are hollow and unbranched, about 25 nanometers in diameter, and are present in structures like cilia, flagella, centrioles, and basal bodies. Overall, these components work together to give the cell its shape, support, and ability to move. Now, let's take a closer look at the functions of these cytoskeletal elements. Starting with microfilaments, they assist in the formation of pseudopodia, which are those temporary projections that help cells move and capture particles. They also form the contractile systems within the cell, playing a crucial role in processes like cytokinesis and cytoplasmic streaming, where they help distribute nutrients and organelles. Additionally, microfilaments provide support to the plasma membrane, maintaining the cell's shape and enabling flexibility. Moving on to microtubules, these structures facilitate the anaphasic movement of chromosomes during cell division, ensuring that each daughter cell gets the correct number of chromosomes. They also contribute to the shape and rigidity of the cell, providing it with structural support. Microtubules are essential components of the cytoskeleton in cilia and flagella, where they enable movement. Lastly, they are involved in forming the spindle apparatus and astral rays, which are critical during mitosis for chromosome segregation and cell division. Overall, both types of cytoskeletal elements are vital for maintaining cell integrity, enabling movement, and supporting cellular functions. Let's take a closer look at the structure of an animal cell. As you can see, animal cells are made up of various key components, each with a specific function that helps the cell operate efficiently. The nucleus, for example, acts as the control center, housing the genetic material and directing activities within the cell. Surrounding the nucleus, you'll find the cytoplasm, which is a gel-like substance where many of the cell's processes occur. The cell membrane is also essential. It acts as a protective barrier while regulating what enters and exits the cell. Moving outward, you'll notice structures like mitochondria, which are often called the powerhouses of the cell because they generate energy. Other important parts include the endoplasmic reticulum and Golgi apparatus, which are involved in protein synthesis and transport. Understanding these structures helps us see how an animal cell functions as a complex, yet highly organized, unit, connecting back to the overall theme of how living organisms are built from these fundamental biological units. Let's take a closer look at the comparison between plant cells and animal cells. Both types of cells have some common features, such as the cell membrane, nucleus, nuclear membrane, and cytoplasm, which are essential for their basic functions. However, there are some notable differences. For instance, plant cells have a cell wall, which provides additional support and protection, whereas animal cells do not. Another key difference is the presence of plastids in plant cells, which are responsible for functions like photosynthesis. These are absent in animal cells. Additionally, vacuoles differ in size. Plant cells contain a smaller vacuole, while animal cells have a larger one. These differences highlight how each cell type is specialized to perform its specific roles within the organism. Understanding these distinctions helps us appreciate the unique features that enable plants and animals to thrive in their environments. Now, as we come to the final slide, let's look at how all these levels of organization come together to form a living being. When different tissues, like muscle or nerve tissue, work together, they form an organ, such as the liver or kidney. These organs don't function in isolation. They coordinate within organ systems, like the digestive or circulatory systems, to carry out complex tasks necessary for life. At the core of all this is the cell, which is the smallest unit of life. It might seem simple, but cells are incredibly diverse and specialized, enabling the formation of tissues, organs, and entire systems. So, to sum up, from the tiny cell to the entire organism, these levels of organization allow living beings to grow, function, and survive. It's this beautifully interconnected hierarchy that makes life possible, and understanding it helps us appreciate the complexity and harmony of living things.